My name is Johannes Verlier, and I call you welcome to the fourth joint working group meetings of Combar. After 3 successful meetings in Warsaw, Leon, and Ghent, we're having our meeting online, as we are all aware due to the circumstances. We understand it's not ideal for in-depth scientific discussions or for making new contacts with collaborators. However, it also offers new opportunities. So to reach out to people, maybe other than scientists from working in the field, or to other members who cannot otherwise physically attend, to be here with us. And I see we are already over 120, so I'm glad you are here with us in such large numbers and from such a lot of different countries. We're over halfway in our project, so it is time to see where we stand and see how the new knowledge that we gain through exchange to new research and to new collaborations, to start to transferring that into for dissemination, but also to see if we can start the process of defining recommendations. Therefore, I look forward to your inputs also in this meeting, so that the more inputs and from the more different angles we can collect inputs, this will help us in defining a view and in defining recommendations that can incorporate all these different perspectives into the topic of enthalmentic resistance. I wish you an enjoyable meeting, a useful meeting, and I hope we will be able to reach out to you in real on future occasions. I'm very happy to give the word to Professor Laura Rinaldi, who is a working group leader 1 and who will share session 1 on diagnosis.
Thank you very much, Johannes. It's my pleasure to open this fourth joint Combar working group meeting with the session 1, Improving the Diagnosis. I am Laura Rinaldi from the University of Napoli, Federico II, and I welcome everybody. We have a 2-hour tight program. We will start after my brief introduction with an invited lecture by Professor Cedric Nouveau from INRA, followed by 3 short presentations by young Combar members, 4 1-minute poster presentations, and finally a moderated discussion with contributions from the panel members and your contribution through Mentimeter. The general aim of our Combar cost action is to combat enthalmentic resistance through an integrated approach in order to have a sustainable control program. And within this approach, diagnostics should not be overlooked. I have been coordinating working group 1 together with my colleague and friend, Professor George von Samson in Mestirna, already for 3 years. We also interact with the other 2 working group leaders, Edwin and Eric. The general aim is to prioritize, evaluate, and implement cost-effective methods for the diagnosis of helminth infections, primarily gastrointestinal nematodes and fasciolepatica, and enthalmentic resistance. We know that the availability of efficient, reliable, and robust diagnostic methods has a pivotal role in order to harmonize study design and interpretation, and also to standardize field and laboratory surveys in different geographical areas in order to achieve an accurate picture of AR in ruminants across Europe. Working group 1 includes 4 tasks. The first is harmonized recommendation for fecal egg count reduction test, a list of available diagnostics and technology readiness level across Europe, market analysis for test introduction, and finally mapping the extent of enthalmentic resistance in Europe through field surveys. And within these tasks, we have been also fostering capacity building by organizing a training school on the advancement of traditional molecular diagnosis held in June 2019 at Cremopar in southern Italy. We know that technology is advancing and it is likely that in a few years' time, novel cutting-edge diagnostics will have been produced and hopefully will be available on the market. These tools should include fecal egg count reduction pen-side tests and associated decision support tools, molecular tests for detecting AR against all major drug classes, as well as in vitro assays, and hopefully also biomarkers to be detected in milk or in saliva. But the tool should meet key practical needs in order to be included in the toolbox for practitioners and farmers. Ideally, a Combar diagnostic test should be convenient to be used on farm, providing results on mobile devices, biodegradable and automated in order to have reduced times and costs. And this will allow us to translate research to practice. We know that automated fecal egg count methods have been developed as the FECPAC G2 or are in the pipeline as our cubic flow-tac microscope. These automated fecal egg count methods will reduce human errors and time of reading. And we ought to aim at tuning automated identification and counting of helmintechs based on artificial intelligence. The general goal is to develop a precision parasitology approach for smart farming solutions. But in parallel, also molecular methods have been developed, especially for detecting benzimidazole resistance with an increasing range of genetic markers. And it seems that molecular tools as pyrosequencing and digital droplet PCR are low-cost approaches for field testing and routine diagnosis. And several collaborative studies are ongoing within Combar on molecular analysis of DNA extracted from larval pools of ruminant samples collected in different countries as Austria, Germany, Portugal, and so on. But whatever test we use, it's important that it is used in the countries because we have to tackle the uptake of diagnosis by practitioners and by the farmers. This is why we have recently run and completed an online survey in 20 European Combar countries in order to identify the opportunities, the barriers, challenges that might affect their commercialization in Europe. And we chose Mini-Flow-Tac as a diagnostic tool, and we performed a SWOT and pestle analysis in order to identify political, economical, social, technological, environmental, and legal aspects that may influence the uptake of these diagnostics. Another task is mapping the extent of enthalmentic resistance in ruminants in Europe. And a few days ago, this nice paper has been just published. We launched a survey, an online survey, and created an open database collecting publication on enthalmentic resistance in gastrointestinal nematodes and liver fluke, representing over 500 studies in 22 countries. And you can see that compared to 5 years ago, enthalmentic resistance is widespread, and so it's really an increasing problem across Europe. From these graphs, you can see that there is an increasing trend over time, especially for benzimidazole resistance, and enthalmentic resistance prevalence is increasing not only in small ruminants as before, but also in cattle, especially for macrocyclic lactones. And from these nice charts, you can see how at least for gastrointestinal nematodes in sheep and goats, the level of treatment failures correlates with the research efforts. So really, if you look for enthalmentic resistance, you will find it. So many actions have been already accomplished, but we have still some activities in the pipelines for next year. First, a market analysis for diagnostics other than fecal egg count reduction test, and potential candidates are ELISA saliva and molecular methods, and as droplet PCR and pyrosequencing. In addition, we are thinking also on a survey on availability of enthalmentics for ruminants across Europe, with a particular focus on prescription situation, which can vary among the Combar countries. In addition, we have already drafted fecal egg count reduction test protocols, circulated and received feedback from many participants, and this is why, in order to finalize these protocols, we have already set up a workshop discussion early next year. Of course, due to the COVID restrictions, many field surveys foreseen for this year have been postponed, but they have been already planned for next year. And as Johannes mentioned, we want not only to do research, but to translate our research output into practical recommendation for the field.
Okay, so yeah, I'm very pleased to now introduce my dear colleague and friend, Dr. Cédric Neveu. He is coming from France, and initially he was trained as a plant parasitologist, but then he found that it's much more interesting to work with parasites from animals, and so he moved into the direction of parasitic nematodes of animals when he obtained a permanent position at the Institut National de Recherche en Agriculture, Alimentation et Environnement, or in French, in English, the French National Institute for Agroeconomical Research in the lovely Loire Valley. And he and his group are well known for performing very nice, elegant studies on the mode of action of enthalmentics, particularly, but not only, on nicotinic acetylcholine receptor agonists or antagonists in parasitic nematodes, but as well on the molecular mechanism of resistance. And yes, I'm very pleased that with this now also his group is somehow becoming a associate with Combar, and we very much look forward, Claude, now to your talk entitled Monitoring Enthalmentic Resistance from Phenotypic Assays to Molecular Markers.
Many thanks, Georg, for the introduction, and many thanks to you and Johannes for the kind invitation to talk to you today about the molecular markers to phenotypic assays. So this work's performed in our team that is mostly dedicated to the search of mode of action and also search for new targets for the development of new drug. But I will give you some example about how our research could be also useful to identify some molecular markers. So I would go very quickly concerning the context, because as Combar participants, you are all aware about the importance of enthalmentic resistance, and Laura made a very nice introduction concerning the rather low efficacy now of BZ and also for macrocyclic lactones, and in lesser extent to cholinergic enthalmentics such as levamisole, parental, and morantel. So the first things I would like to address is why do we need these molecular markers? First, they're going to be very useful in order to optimize the enthalmentic treatments. They can be used also as predictive tools for predictive potential cross-resistance, and also they can be very useful to monitor spreading of allele resistance into the population. So this is not an exhaustive list, but at least these points are very important. I will try to give you some example about how we are identifying these molecular markers for enthalmentic resistance using either candidate gene strategy based on C. elegans data, or using without a priori approaches such as comparison of worms that are susceptible or resistant to enthalmentics. And these comparisons are performed at the genomic or transcriptomic level. And finally, we will see how to validate them both in the lab and in the field. So surely I have only 15 minutes or something like that. So I will really focus my talk today on one class of enthalmentics that are the cholinergic enthalmentics, the one that remains potentially efficient to control both benzimidazole and ivermectin's resistant worms. And I will focus on levamisole, parental, and morantel. So first of all, how do these drugs are acting on the worm? So these enthalmentic compounds, such as levamisole, parental, and morantel, but also a lot of them, such as oxantol, nicotine, dercontel, and so on, are activating or inhibiting the acetylcholine receptors from the worms. These receptors are pentameric ligand-gated ion channels that are involved in fast synaptic neurotransmission in both vertebrates and invertebrates. And their permanent activation or inhibition by the drugs will lead to the worm paralysis. And then the host perfectly knows how to get rid of the paralyzed worms. So you can have two different kinds of receptors. The first one are homopentamers. They are made of the same subunits. And the other one can include combination of different subunits, and they are the heterocontaminant receptors. What is important to know here is that different subunit combination represent distinct subtype of AChR, and that the different enthalmentics can be selective for these different subtypes. So here, our working hypothesis was very simple. Because the best molecular markers are directly involved in the phenotypes, we decided to investigate polymorphism that could impact the functionality of these receptors that are targeted by the enthalmentic drug. So I'm sure that most of you perfectly know that game that consists on identifying a combination of 5 different colors. And clearly, we are facing a problem with nematodes because there is a large diversity of AChR subunits in nematodes, much more than in vertebrates. So for example, there are at least 29 different subunits in C. elegans in nematode model and 30 in Emanchus contortus. So clearly, if we had to identify the enthalmentic targets just by testing random subunit combination, it would be like playing a super mastermind game with more than 30 different colors. So clearly just impossible. So fortunately, we have here 2 main models that could help us to identify the molecular targets. The first one is C. elegans. So C. elegans, the good news is that this model nematode is susceptible to all cholinergic agonist enthalmentics. And clearly screens for mutants that survive the enthalmentic exposure have been of major help in order to identify the AChR subunits that are involved in the corresponding receptors. However, clearly, there are some also major limitations with this model. First, C. elegans is not a target species, and clearly AChR subunit combination and pharmacological properties of his receptors can be different from the one from the parasites. So we also have the frog's eggs. This frog is an African toad, and this egg is a wonderful model for electrophysiology experiments. It is widely used to express nematodes AChR receptors made of AChR subunits. And by using the 2-electrode voltage claim techniques, you can record some currents that are elicited by acetylcholine, but also the cholinergic agonist. Here you've got an example of a recording trace. So the receptor is expressed at the membrane, and when you apply acetylcholine, you observe this depolarization of the eggs, And the same thing with levamisole, showing that you have expressed the levamisole receptors into the eggs. So this model is extremely useful to decipher the subunit combination of the nematode AChR, but also to perform detailed pharmacological characterization of the receptors, and surely to investigate the role of the polymorphic AChR subunits that are potentially involved in enthalmentic resistance. So let's start with the candidate gene strategy. So in C. elegans, the selection of mutants that survive exposure to levamisole has led to the identification of 5 different genes encoding 5 different NAChR subunits. And these subunits are called UNC38, 63, LEV8, UNC29, LEV1. UNC is for the name associated with the loss of function of the gene. UNC is for uncoordinated, meaning that the worms that lacks these subunits doesn't move normally. LEV is for levamisole resistance. And what has been shown by Thomas Boulin in 2008, so an old paper, that if you express these 5 subunits together into the Xenopus oocyte, then you get a functional receptors that is sensitive to acetylcholine and that is responsive to levamisole. So demonstrating that these receptors potentially is the levamisole receptors in the model species. And also very importantly, it has been reported that UNC38, 63, and 29 null mutants are resistant to levamisole, parental, and morantel. So clearly suggesting that these receptors could be the main targets for the 3 different drugs into C. elegans. And these data laid a strong basis for the candidate gene strategy. And this is why we try to search for homologs of these genes into the parasites and try to identify putative polymorphism associated with resistance. So the first thing to address is that, was it possible to have a direct transposition of these C. elegans data to parasitic species? So orthologs of UNC38, 63, and 29 can be easily found in all nematode clades. And with the genomic research we have right now can confirm that these subunits are highly conserved all through the nematode's phylum. Whereas in sharp contrast, there are no homologue of LEV8, but also LEV1 in some case, in many parasitic species that are sensitive to levamisole, parental, and morantel. So this clearly suggested that LAChR might have different subunit combinations between C. elegans and the parasites. But at least as starting points, we started to compare what we had into our hands, meaning 38, 63, and 29 in levamisole susceptible and levamisole resistant isolates from Emonchus, Deladosagia, and Trichostrongylus. So just by performing simple RT-PCR on cDNA from the 3 species, from levamisole resistant and levamisole susceptible isolates, what we've been able to show is that the complete coding sequence are expressed in both levamisole resistant and levamisole susceptible isolates. That when you look carefully at the sequence, you don't find any polymorphism or changes between levamisole resistant and levamisole susceptible isolates. But what we observed is that in our highly resistant isolates from Emonchus, Deladosagia, and Trichostrongylus, we identified an additional amplification product. So then we cloned it, and what we found is that this additional amplification product observed in the 3 species corresponds to a spliced variant of UNC63. And for Emonchus, we have raised some antibodies that are against the peptide that is common to both the full length and the truncated isoform. And we've been able to confirm in the levamisole resistant isolates that you have the co-expression of the full length mature subunits, as well as a truncated isoform of the UNC63 gene. So at the end of the candidate gene strategy, what did we have into our hands? So first we had at least 3 potential LAChR subunits from 3 different Trichostrongylus species. And we have observed a polymorphism here that corresponds to the co-expression of both complete and truncated UNC63 protein in worms from highly resistant isolates. So this was a good starting point, but clearly it was very incomplete. So we needed for a complementary approach in order to identify other markers, and if possible, some markers that could be common to resistant isolates and highly resistant isolates. And also we need to identify the other potential AChR subunits that are involved in the LAChR composition from the parasites. So what we did here is that we decided to perform a without a priori approach, where the main objective was to compare the transcriptome from levamisole resistant and levamisole susceptible Haemoncus using cDNA FLP. That is a molecular biology approach that is very, very attractive because it will allow you to investigate expression polymorphism, but also transcript polymorphism, such as point mutation and insertion and deletion events. So we performed these experiments using 2 resistant isolates and 2 susceptible isolates. And among the 17,000 transcript-derived fragments that were generated, only 26 presented a polymorphism between resistant and susceptible nematodes. And 15 of them were present in both susceptible isolates and absent from both resistant isolates. But even more attractive, 11 were present in both resistant and absent from the susceptible isolates. So potentially some very attractive resistance marker here. And among these attractive candidates, we've been very pleased and lucky to identify a fragment that is called HCO-RCR8B, because this corresponds to a spliced variant to HCO-RCR8 that is specifically expressed in levamisole resistance, and RCR8 is an NSRHR subunit that is closely related to LEV8. So suggesting that RCR8 would potentially replace the missing LEV8 subunit into Haemoncus contortus, but also other nematodes levamisole receptors. This was also a very important result because we've been able to confirm that this spliced variant from RCR8 was also found in other isolates from our lab and specifically expressed in the levamisole resistant and absent in levamisole susceptible isolates. But this was also confirmed since by other labs on many other field isolates from Australia, North America, South America. So a nice and promising marker. And also very interestingly is that as observed for Un63, the abbreviated isoform from RCR8 was co-expressed with its full length in levamisole resistant isolates. So clearly this was suggesting that the expression of some abbreviated isoform of the AChR subunit could constitute a response to the selective pressure exerted by the enthalmentic. And this is also further supported by the work performed on monipental, where Lucien Ruffner in Ronald Kaminski group showed that Haemoncus also responds by expressing truncated isoform of HCO-MPTL1, which is the target of the monipental in Haemoncus. And this was also recently confirmed in field isolates by Andrew Coetzee on Australian isolates. So now the next step is how can we investigate the putative involvement of this truncated isoform spliced variant into resistance? So here, let's go back to the Xenopus oocyte. What we did first is that we tried to co-express into the Xenopus oocyte the subunits that we had into our hands, 38, 63, RCR8, and Un29. And we've been very lucky at getting a very nice and robustly expressed functional receptors here that is extremely responsive to levamisole. Here you can see that levamisole is acting as a full agonist on these receptors. So levamisole is more potent on these receptors than the C. elegans receptor. And clearly, it was for us also a major opportunity to test the role of the truncated Un63. And by mimicking what we can observe into resistant worms, we co-expressed in the Xenopus oocyte all the subunits that are required to get the functional expression of the levamisole receptors, in addition to 63 at different ratio, 1:4:2 and 1:10. And what you can observe here is that the expression of the receptors is severely impaired when you had this truncated isoform. And this strong dominant negative effect on the receptors was something very important because it was representing at that time the first functional evidence for a potential molecular mechanism of levamisole resistance in parasitic nematodes. So then surely we tried to do the same thing with the truncated HCO-RCR8 by co-expressing truncated HCO-RCR8 with the levamisole receptors. But here, unfortunately, we didn't get any results. We didn't observe the dominant negative effect. Potentially, we hypothesized that's because there is a lack of transmembrane domain, and that potentially also xenopus might not be the best model to test the involvement of RCR8B in resistance. But clearly, what is important to have in mind is that the role of the truncated RCR8 in levamisole resistance remains to be established. So anyway, we were not able to play with the short one. So we tried to get some more information about the full length one, about RCR8. What is the role of RCR8 more precisely? So when we tried to identify the minimal subunit combination that give rise to the functional receptors, what we noticed is that when we got rid of RCR8 in the mixture, we completely lose sensitivity to levamisole. We make a new receptors. We make a new receptors that is able to assemble without RCR8, and this receptor is far less sensitive to levamisole. But in sharp contrast, this receptor was highly sensitive to parental. That was very, very attractive because clearly, because the difference between H-co-LCHR1, this one, and H-co-LCHR2 is the only presence or absence of H-co-LCHR8. So this clearly highlights the critical role of RCR8 into levamisole sensitivity. So indirectly, this highlighted the importance of the RCRB marker and also suggested that in sharp contrast with C. elegans, levamisole and parental could act on distinct receptors in Emanchus contortus. So why do we, how can we test the hypothesis of one target versus multiple targets within the worms? So what we did is that we tried to inject RCR8 into C. elegans Lev8 mutants, okay? And what we observed is that when you do that, it does not change the motility of the worms. The N2 are moving normally, the Lev8 mutants are moving normally, and the one expressing the RCR8 from Emanchus are also moving the same way. But when you apply levamisole, clearly the N2, the wild type strand, is paralyzed by levamisole. Lev8 is resistant to levamisole, and the transgenic line Lev8 expressing RCR8 will become sensitive to levamisole. So we restore levamisole sensitivity here. And exactly the same thing for parental, meaning that just by adding one subunit, we were restoring both levamisole and parental sensitivity, suggesting that in C. elegans, levamisole and parental could have the same targets, that is the LSCHR. But strikingly, when we investigate RCR8 in vivo into Emanchus contortus by silencing the subunits into L2 stage, what we observed was something very different. Because when you silence RCR8 into Emanchus, then the worms become resistant to levamisole, as you can see here. Whereas in sharp contrast, this does not affect at all the parental sensitivity. And this is something that is of major interest. Unfortunately, we are not able to silence also RCR8B into our levamisole resistant worms because it was not applicable, as the sequences are too closely related between the short one and the long one. But clearly, these results were supporting the hypothesis that levamisole and parental could have distinct molecular targets in Emanchus. And also of importance, when we were investigating some novel ACHR muscle subtypes in order to get some new targets for the development of new enthalmentics, we identified a novel ACHR muscle subtypes that we call the MSCHR. These receptors is made of two subunits that are only found in animal parasitic nematodes and that are absent in the free-living nematodes, such as C. elegans, but also absent in plant parasitic nematodes. And 2627 makes a functional heteromeric channel, so expressed in the muscle, and this receptor, surprisingly, is incredibly responsive to morantel, and in a lesser extent to parental. But what is very important here is that we got an additional subtype of receptors that is completely insensitive to levamisole. So when we expressed these receptors from Emanchus or from the distantly related parasites, Parascaris equorum, into C. elegans, we were able to show that expression of this MSCHR into C. elegans was leading to a drastically increased morantel sensitivity to the worms, and in a lesser extent to parentel, so validating this novel muscle subtype as the morantel AChR, MSChR. So then, with all this information into our hands, how can we speculate about the interest of truncated UNC63 or RCR8B as marker for predictive, as a predictive tools? Here I would say that it might not yet be the case. Okay, so we got the HCO-LCHR1 made of 38, 63, RCR8, and 29. We've been able also to show that these receptors was clearly obtained also in other species, such as Oesophagostomus dentatum. We've got, and these receptors is supposed to be the main targets of levamisole. We've got the second subtypes, HCO-LCHR2, that is also found in Oesophagostomus dentatum, the one that is lacking RCR8, and this one might be a preferential target for parentel. And we've got this third subtypes made of 26, 27, that has been also confirmed in distantly related parasites such as Parascaris equorum. And this one might be a preferential target for morantel and parentel. So now concerning the truncated UNC63, if we assume that the AChR that includes UNC63 could be impacted by the truncated isoform, then we could predict a cross-resistance to levamisole and parentel if we identified UNC63 into a worm, truncated UNC63 into a worm. And this is what we confirmed in vivo in the lab, selected HCO-RHS6 that comes from Marlene Ross labs, that has been selected artificially for its levamisole resistance starting from a susceptible strain. So this strain has been selected only for levamisole resistance, have never been in contact with parentel, and we demonstrated that in vivo the receptor, the worms are cross-resistant between levamisole and parentel. So clearly now this has to be further confirmed in field isolates from Emonchus, but also as we also identify this truncated UNC63 in highly resistant isolates from Teradorsagia or Trichostrongylosis, this has to be checked also for this species. We have also to confirm that this truncated UNC63 is strictly associated with high level of resistance. And this is also of importance as this cannot be considered as a universal marker. Now concerning RCR8B, even though it has been associated with levamisole resistance in many field isolates by different labs, there is currently no functional validation for these markers. But at least what we can do is to investigate potential RCR8B in other levamisole resistant from other species to see if we are keep going on confirming that this might be a marker of interest. But because levamisole, parentel, and morantel could have differential or preferential targets into the worms, depending on the history of the treatments, this also means that there is potentially still a chance for parentel or morantel to remain active on levamisole-resistant parasites. And clearly what it suggests is that instead of monitoring enthalmentic resistance as the only goal, we should really start thinking about also monitoring enthalmentic susceptibility. Because levamisole, parental, and morantel side resistance have been described in field or laboratory isolates from many species. Clearly, it was commonly accepted that resistance to these cholinergic agonists was co-inherited. But in sharp contrast, our results suggest that levamisole, parental, and morantel cross-resistance might not be inevitable. So, supporting this hypothesis, other studies have already reported that in field or lab-selected isolates of at least Trichostrongylosis, morantel-resistant worms remain sensitive to levamisole. And in our lab, in some of our own strains, we showed that some multi-resistant worms that are resistant to benzimidazole, ivermectin, levamisole, and parentel remain susceptible to morantel. And they are also accumulating evidence for potential levamisole reversion. In some of our strains, if we do not maintain levamisole pressure, selective pressure on these worms, they come back to levamisole sensitivity. So clearly now what we need is a tool to monitor at the same time both susceptibility and resistance to levamisole, parental, morantel, but also surely all the other enthalmentics in a wide range of field isolates. And in that respect, what we would like to do is to take advantage of the automated larval migration assays that have been recently developed into our lab by Fabrice Guignard. So this technology was first developed for quantifying RNAi phenotype on M. emonchus contortus. So this technology is based on a spectrophotometric approach that is used for the monitoring of the larval stage motility. And this is real time. Here, just some example of the curves you can get from a susceptible isolate and a multi-resistant isolate. And here in black, I mean, this is the accumulation of fluorescence that is correlated with the migration of the larvae into the recording chamber. And you can see that you can detect slight difference of motility depending on the dose that you are using. And just to give you an example here, with parental with 0.75 micromolar, you're almost completely paralyzed. Whereas to get the same phenotypes, you need to use 64 micromolars for parental. And the good news is that this is also working for ivermectins. And here for 0.02 for the complete paralysis, here for the complete paralysis, you need 0.32 micromolar. So this technology is very attractive because it can allow you to estimate quite quickly the EC50 and also the resistance ratio, here, for example, for parental. But it's also very attractive because it can allow you to identify some active compound to control the resistant worms. And for example, here are some drugs that are as efficient on both the susceptible and multi-resistant strain. So what we are doing right now is that we have already successfully adapted this technology to a wide range of enthalmentic drugs, including MLs, so that is very important. But also we have adapted it to a wide range of parasitic species from the different clades, and it also works on plant parasitic nematodes. And what are the work that are ongoing? Now we are trying to use this assay to detect resistance into mixed species that is more relevant with what we found in the field. So clearly, it is going to be very useful to evaluate the robustness of the different molecular markers we already have into your hand and the one that will come in the next future. But this will be also a very attractive decision-making tool for the field in the near future, something that will be very complementary to the fecal count reduction test. So to finish, I would like to acknowledge most of the present and past members from our team, as well as all the collaborators that are involved in this project. And I would like to thank you for your attention. And if I can answer your question, it would be my pleasure. Many thanks.
Thank you very much, Cedric. Excellent talk. And I can see from the Mentimeter some questions. So we start with the most voted one. How do you suggest to monitor efficacy and resistance in the same time?
So the answer is quite simple. At the starting point, I mean, we were using, single runs of larvae. So you had one batch of larvae, one drug, and you were testing, okay, how does it migrate? And then you get your answer. It takes something like 15 minutes. Now the test has been adapted to work with high throughputs. So we are able to perform based on the collaboration with Invenesis. The technology has been adapted to 96 and even 380 well plates. So at the same time, you have your batch of larvae. You will spread helicots of your larvae on all the plates, and you would just need to distribute different concentration of the different enthalmentics. And you will have your controls, susceptible worms, resistant worms, and you will exactly see how they behave, and you will be able to determine with the same sample which drug is remaining active on the worms and which drug is not active anymore. And this will be very informative because we will be able to detect potential cross-resistance, and you will be able also to help the vet for the decision by saying, okay, you've got this resistance, but your worms are still susceptible to this drug. So maybe it might not be applicable because you might have some constraint because of your lactating animal or something like that. But at least you can have a good overview. And because it has been adapted also to high throughputs, you can also imagine that you can make this diagnosis at the ship level, because you only need something at about 300 to 400 larvae per run in the plates. So if you got enough fecal material, you will have enough larvae from a single sheep to describe all the sensitivity and all the resistance at the single animal level. That would be also very useful. Or in contrast, you can think about pooling your fecal sample and have a rough idea about what is occurring in one place.
Okay, so we move to the first short presentation by Kira Hamilton from Tegas.
So, good afternoon. Thank you for inviting me to present my work here. I'm Kyra Hamilton, and I study my PhD with UCD Dublin and conduct my research between Chagaskin Island and Ag Research in New Zealand. Today, I'm going to focus on the paper, Survival of the Fittest, Ecological Fitness in Telodosargia circumcincta of known enthalmentic resistance status. Now, the background for this project lies with nematode parasites of sheep, primarily with Telodosargia circumcincta. I'm sure you're all aware of the importance of parasitism as a production limiting factor for grazing livestock. And most of you will also be aware that the long-term reliance on enthalmentic drugs to control these worms has resulted in the escalating issue of resistance among the worm populations. Now, a great deal of money and effort has been invested in finding ways to slow the development of enthalmentic resistance to these important drugs. And this is essential to ensure the sustainability of our industry. Now, several years ago, something really interesting and perhaps a little surprising happened in some on-farm studies conducted in New Zealand. So what you can see in this graph here is that when a complex of resistance management strategies were applied to a series of commercial sheep farms, all of which had pre-existing issues with enthalmentic resistance. And over the course of 5 years of monitoring, there was an overall improvement in the treatment efficacy against Telodosargia circumcincta. Now, this occurred in all 3 of the older enthalmentic classes, so our classic BZ, levamisole, and macrocyclic lactones. So this observation raises a question as to the possibility of managing our way out of a resistance problem once it has developed. So my PhD is studying and aiming to try and understand one possible mechanism for this to occur. So what we're going to look at is fitness. So the hypothesis we came up with was, are resistant mutations resulting in ecological fitness costs in Telodosargia circumcincta? Now, fitness is defined as the ability to survive and reproduce. In internal parasites, it therefore involves establishment in the host, reproductive ability, infectivity, and lifespan. So developing in vitro assays to quantitatively measure aspects of parasite fitness required assessing traits deemed essential for the life cycle. So for Telodosargia circumcincta, this included measuring fecundity, migration after exposure to hot temperatures, egg hatch percentage after exposure to cold temperatures, egg hatch under regulated temperature, and exchievement from free living to parasitic stage 3 larvae. Now, 4 isolates of known enthalmentic resistant status were used. So isolates D and M were multi-drug resistant, and to save confusion here, I'm going to refer to them as R1 and R2. And then we also have isolates T and W, which were pan-susceptible. And again, to save confusion, I'm going to refer to them as S1 and S2. So R1 and R2, resistant 1 and resistant 2, and S1 and S2, susceptible 1 and susceptible 2, as you can see from my table here. So in total, 8 lambs were infected, 2 for each isolate, and they generated material for the in vitro fitness assays. A larval migration inhibition assay was used to confirm the resistant status of the isolates against ivermectin. And from this, R1 and R2 were confirmed resistant, and S1 and S2 confirmed drug susceptible. Now, there were 5 assays selected to quantify aspects of parasite fitness. So we used larval length as a measure of fecundity, but we also know from other research that multi-drug resistant Telodosargia circumcincta are shorter than their susceptible counterparts. An egg hatch assay at regulated temperature was used to measure the basic hatching ability of the isolates. We also looked at the percentage exchievement from 3rd stage free living to parasitic larvae, and we did this using the exchievement assay. And after this, we measured the migration ability of 3rd stage larvae and this involved exposure to hot temperatures for a set length of time, and this was called the accelerated aging test. And then finally, the cold stress test measured the percentage development of egg to third stage larvae after exposure to cold temperatures for a certain number of days. But what were the results? So after initial analysis, the assays turned up some interesting results. The larval length assay assessed the lengths of 100 different larvae for each isolate and revealed that S2, so the susceptible to isolate, was found to be significantly longer than all other isolates. The egg hatch assay revealed that isolate R2 had a consistently higher hatching percentage than S2. So in this table here, all the values with the same superscript had no significant differences. If there's a different superscript, there's a significant difference. And then we followed on with the exheathment assay. So for the exheathment assay, larvae were incubated in artificial rumen fluid for up to 24 hours before the percentage exheathment was determined. Now, from our results, and in the box I've highlighted in red here, this is where our significant results occurred. Isolates R1 and S1 had a higher percentage exheathment than that of isolate S2 after 24 hours. For the cold stress test, similarly, I've plotted a box plot. So the fecal samples here were incubated at 4 degrees Celsius for various lengths of time. And after this, the percentage development was determined and the initial time point was used as a base level. So time point 0 was used as 100% for the hatching percentage. Again, here I've highlighted in red the most significant area where the results were found. The area with the most significant results were found. So at time point of 4 days, for the cold stress test, this revealed differences between isolates R1 and R2 and S1 in their egg to stage 3 larvae percentages. Again, the accelerated aging test. So interestingly here, we looked at third stage larvae migrating after incubation at 30 degrees Celsius for various lengths of time. So as you can see, we have hours extending from 0 to 504 hours of heat exposure. So the results of the migration following the accelerated aging assay showed that all the isolates displayed a reduction in their migration ability over time. In particular, the only significant differences were seen between isolates R2 and S1 and isolates R2 and S2 at time point 504 hours of heat exposure. Again, I've highlighted it with a red box because there's a lot of information happening here. But what is so interesting about these results? So these results don't specifically give us a definitive answer as to whether or not resistance to one or multiple drugs results in a fitness cost. But what it does show is that there are fundamental differences between the base and or background level fitness between the different isolates. Apologies, I'm just going to go back a slide. Yeah, so fitness doesn't necessarily correlate with resistance status, but with the wide variation in background fitness, it does suggest that in order to prove the hypothesis or disprove the hypothesis, we need to do more research, which brings us on to my current studies. So currently, there are studies ongoing to investigate the relationship between resistant genotypes and fitness in a single population. So for this, I have used a single isolate population and split it into its different subset known multi-drug, and we've done this using multi-drug resistant populations that we know are multi-drug resistant. And in order to split the population, we have used a different combination of enthalmentic drugs. So we've used unselected single, double, and triple combinations for this. And through using this single isolate population, so we hope to reduce the ecotypic variation as happens when you're comparing multiple different isolates from different sources. And also we hope to minimize the background variation in fitness as we've seen from this current trial that I'm doing. So in summary, there are a large variation in the background fitness of different isolates. And the hypothesis that I have worked on cannot be conclusively accepted or rejected from this trial work. However, that's why further testing is underway, and this work will hopefully work towards helping me being able to conclusively accept or reject the hypothesis of the work. Thank you.
Thank you very much, Kyra. Very interesting. Okay, I don't see any question from the Mentimeter, but maybe I have one. So those isolates were from sheep, isn't it?
Yes, so my work is in sheep parasites.
And do you think that this kind of ecological experiment can be applied also to other gastrointestinal nematodes, or are you planning to do other studies?
Currently, my studies are only in sheep, but if we look at the specific life cycles of other parasites, we could adapt it similarly towards them. I think it's important to look at aspects that are essential for their life cycle, like, you know, with liver fluke, you have specifics towards them where they migrate into the liver, and the most damage is caused during the juvenile stage, during their migration.
Oh, there is a question. Okay. Have you considered collecting early and late HX and then testing whether the larvae have the benzimidazole SNPs?
SNPs, single nucleotide polymorphisms.
Yeah.
I don't do the molecular work, but so I don't know specifically about that, but it would be interesting in the future to look at collecting between early and late in the year so that we could compare the fitness then, because that's part of the work I'm looking at with the cold stress test and the accelerated aging test. It puts them under heat and cold stress to try and emulate what would happen if they were put under such stresses at different times of the year in a fake environment in the lab.
Okay, thank you very much, Kyra. Thank you.
You.
And the next speaker is Nadine Ravinet from Oniris in Nantes, France.
Okay, thank you. So my presentation is entitled Bull Tank Milk Osartaja ELISA as a tool for quantification of milk production losses in dairy herds. So the question is, where do we stand? So you will see this presentation is totally different from those regarding molecular markers, for example. To rationally control gastrointestinal nematode infection, we want to make sure that we do not treat for nothing. So there's an important need for tools that correctly predict the level of loss or the gain to be expected from treatment. In the particular case of grazing dairy cows, we know that this infection can induce milk production losses. And there's a very easy to use tool available on the field. This is the Osartaja ELISA, which measures the bull tank milk Osartaja antibody level with results expressed as optical density ratio, ODR. So does this tool do the job? The objective of my presentation is to provide some answers to this question based on the results obtained over the last 15 or 20 years. So initial work focusing on this bull tank milk Osartaja ELISA showed significant negative statistical associations between ODR values and the average level of milk production. So you can look at this summary table, and we can see that for those different studies, when the ODR value increased from the first to the third quartile of the distribution within the dataset, then the associated decrease in milk production was around 1 kilogram per cow per day, this result being consistent across the various datasets. Moreover, all those studies had the advantage of having been carried out in large to very large study sample, as you can see here, and in commercial dairy farm, which are representative of the field. However, those significant negative statistical associations did not demonstrate a causal link between gastrointestinal nematode infection and reduced milk production. So the question is, in high ODR herds, is the lower milk production necessarily and only due to Osartaja infection of cows? In fact, several other factors could contribute to this negative relationship between ODR and milk production. And you have here some example of different factors which affect at the same time ODR value and milk production without establishing any causality. So when questioning this causality, we can wonder what could be the expected increase in milk production after enthalmentic treatment, especially in high ODR herds. So we can look at other studies assessing the milk production response after enthalmentic treatment according to different ODR values. So we can look again at this summary table where milk production responses are compared between high ODR herds and low ODR herds. First of all, in those two studies carried out in Canada and France, we can see that the milk production responses were not significantly different between high and low ODR herds. But this study was carried out in herds with limited outdoor exposure, and this one with a very small study sample, only 25 herds. On the contrary, here we have a study where the effect of treatment was significantly higher in high ODR herds, but after this great effect, the next greatest effect was obtained in the opposite category of herds, that is to say in herds with a very low ODR. And in this study, the effect of treatment was negative. That is to say that we observed a decrease in milk production after enthalmentic treatment, this decrease being significantly higher in high ODR herds. And in this last study, several thresholds of ODR values were tested on the same dataset. We can see here that the threshold 0.9 could be seen as interesting, even if the discriminating power was very low. That is to say that the difference between those two milk production responses was not very important. And the relationship between the ODR value and the milk production response was not linear, because as you can see here, for the threshold 0.7 and 1, we obtain exactly the opposite effect of the expected effect, because the milk production response was higher here and here, that is to say, in low ODR herds. So several authors suggested that this predictive value of the bird tank milk ODR might be improved if the ODR was combined with on-farm analysis of management factors. So first, we tried to combine the bird tank milk Ostertagia ODR value with the TEC, that is to say the time of effective contact with L3 before the first calving. And as you can see here, we showed that the milk production response was better in herds characterized by a low TEC and a high ODR compared to herds only characterized by a low TEC, whatever the value of the ODR. And next, we tried to combar the ODR value with the TEC and the percentage of grazed grass in the diet. And you can see here that the best milk production response was obtained in high pasturing herds with a high ODR and a low TEC, compared to high pasturing herds only characterized by a low TEC. So those results suggest that this combination could be very interesting to improve the predictive value of the ODR. But here I would like to draw your attention to the fact that when we combine several indicators, then the number of herds considered in the sample is greatly reduced, so the level of evidence automatically goes down. So, what could be the other limits of those studies assessing the predictive value of the bird tank milk ODR for the milk production response? Those studies were often based on a single ODR value measured at a given time. But we know that the bird tank milk ODR value can vary over time, and particularly during the autumn period. So further questions are, is there an optimal date for bird tank milk sampling and Ostertagia ODR measurement? That is to say, a date with the best predictive power of the ODR measurement performed? Or can this predictive power be improved by combining several ODR values from milk samples taken at different times? So we tried to answer those questions using blood tank milk Ostertagia measured fortnightly from the 1st of October until the date of treatment here. 9 to 7 blood tank milk ODR values were obtained per herd. And thanks to this enthalmentic treatment and those production milk data, we were able to assess the milk production response after enthalmentic treatment according to several ODR values measured at different times. Those several ODR values were taken into account either independently of each other. For example, we consider the ODR at the beginning of October or November or at the time of treatment, or taken into account in combination. For example, we consider the average of several Ostertagia ODR values or observation of several ODR values higher than a given threshold. So 14 ways of taking those ODR values into account were tested, and each time with 4 different thresholds, as you can see here. So finally, 56 variables were tested. And the main result is that none of these 56 variables were able to predict correctly the milk production response after enthalmentic treatment. So to conclude, we can suggest that the blood tank milk Ostertagia ODR cannot alone reliably quantify milk production losses due to GIN infection. In fact, the impact of GIN on milk production depends on at least those 3 factors: resilience to infection, resistance, and exposure to parasites. The blood tank milk Ostertagia ELISA can be a good reflection of resilience or resistance. And regarding the exposure, we can consider that this ELISA could be a good reflection of the herd exposure to GIN. In fact, significant statistical associations were found between ODR values and duration and access to pasture and whole herd treatment. However, in all those studies, when we look at the coefficients of the models, we can see that they are not very high, very important. And we have to keep in mind that Sanchez and Doe highlighted that after controlling for cow exposure to pasture and whole herd treatment, a large amount of the variation of the full ODR was not explained by factors in this study. So even for that factor, we can't be totally sure that the Ostertagia ELISA is a perfect reflection of the exposure to GIN. So thank you for your attention, and if you have any question.
Thank you, Nadine. Thank you, well done. So the most voted question is in the Ostertagia ELISA, is there a cross reaction with the other elements?
Yes. I think that there are some papers showing that there are cross reaction with Fasciola hepatica, and this is one of the reasons explaining why we can question the causality between Ostertagia and reduced milk production, as of course Fasciola hepatica can also induce milk production losses.
Thank you very much. Thank you, Nadine. So Natalie Jacobs.
Thanks at first for giving me the opportunity to present here a part of my PhD project. And within this talk, I would like to show you how hemoncocib that is expressed in the model organism C. elegans can influence the ivermectin susceptibility. So as you are all aware of, Hemoncos contortus is a gastrointestinal parasite infecting small ruminants. And infections with this parasite are mainly treated with broad-spectrum enthalmentics such as macrocyclic lactones. So as pictured here on the right, the most representatives are ivermectin and moxidectin, which act by permanent activating glutamate-gated chloride channels or GABA receptors in the nematode, leading to a flaccid paralysis and finally to death. So, however, the intense use of these drugs has led, as you know, to the emergence of resistance, why discovering the specific resistance mechanisms remains an important challenge today. So what is so far known about specific ML resistance mechanism is that there's no single mechanism. So we have a polygenic nature of ML resistance mechanisms is assumed, which can be divided into target site related and non-target site related ones. However, drug metabolism is by far the most important drug defense mechanisms in all organisms, as you can see by a plenty of examples in humans. So having now a look on if drug metabolism has also an impact on ML resistance in nematodes, especially Hemoncus, Matuskova et al. Reported in 2018 that there is a constitutive expression of a UGT transferase, so a phase 2 enzyme in resistant Hemoncus strains. But biotransformation can include a variety of enzymes, and for that, xenobiotic metabolism is divided into two phases, so phase 1 and phase 2, which are divided because of their according catalyzed reactions. So when having now a close look into cytochrome P450 mediated metabolism, which is by far the largest enzyme family for drug metabolism, and connecting this cytochromes with macrocyclic lactones, only for the human CYP3A4 enzyme, it has been linked that this enzyme can metabolize ivermectin. However, cytochromes in C. elegans hasn't been so far associated to metabolize ivermectin nor moxidectin. And for that, what I'm interested in my PhD is if a single Hemoncus contortus gene that is transgenically expressed in the model organs can have an impact on the susceptibility towards ivermectin or moxidectin. So as I mentioned, there is only less data available on CYPs associated to ML metabolism. And for that, we first had to identify a candidate gene. And we had a look, therefore, in a study from Choi et al., who introgressed Telodorsagia triple-resistant isolate into a susceptible background and performed an RNA-seq experiment. So now having a look into the data, we identified a couple of upregulated candidate genes. But when having a close look onto this specific gene, which is an orthologous to the CYP13A family, it was 20-fold times overexpressed in this resistant Telodorsagia isolate. So there is an orthologous gene also in Hemoncus, and for sure we find one. And I had a look into the literature so far and checked whether this gene or this subfamily of CYPs has been so far associated to be xenobiotic inducible. But it has been not. A study from Lying showed as well that this CYP subfamily is also only low expressed in fourth stage larvae as well as in male and female adult worms. So now to check whether this gene can have an impact on the susceptibility or not, I assembled our candidate gene into a kind of expression plasmid under the control of a GAS1 promoter from C. elegans to ensure the intestinal expression. And what I did then was I co-injected this plasmid we generated with a GFP co-marker into the gonads of the wild type N2 strain, and then checked whether there are GFP positive transgenes or not. So GFP was only used as a marker. It is here expressed in the pharynx and just was used to facilitate selecting positive worms. Afterwards, I conducted thrashing assays, since ivermectin and moxidectin also can have an impact or affecting the locomotion of the worms. So I incubated young adults with either ivermectin and moxidectin at different concentration for 24 hours, and then counted the body bands per minute of single individual worms. So what you can see here are now the resulting concentration response curves. So here the motility in percent is plotted against the drug concentration for either ivermectin or moxidectin on the right in nanomolar in a logarithmic scale. And when having a look onto the curves for ivermectin, we're able to see that there's a right shift when incubating the transgene line with ivermectin compared to the wild type. So this means that we also have a shift in the resulting EC50 values. So in comparison to the N2 with the EC50 value of 1.6 nanomolar, the transgene is showing EC50 value of about 7 nanomolar, which means that there's a fold change of about 4. When now having a look onto the data for moxidectin, we're not able to see any shift in the curves. So no difference between the transgene and the wild type control, leading finally also to similar EC50 values. So what we can say by this graph is that this hemoncocib from a highly resistant hemoncos isolate, white river in this case, and expressed in the model organism C. elegans, can modulate the ivermectin susceptibility, but it does not for moxidectin, which is highly interesting. And to somehow explain this phenomenon that can be observed, I had a close look into the structures. So what you can see here are the superimposed structures of ivermectin in black and moxidectin in blue. In a binding site, so binding in a subunit of a glutamate-gated chloride chains, just to show you the differences in interactions. So what you can see here is that both macrocyclic lactones share the same core structure. However, ivermectin is possessing a big disaccharide group, leading to also somehow slightly different interactions in this glutamate-gated chloride channel. However, moxidectin does not possess this disaccharide group, but instead a metoxime group and a different alkyl side chain. So what I assume now is that there is somehow a specific substrate specificity of this hemoncocib. And to gain further insights, I'm currently conducting thrashing assays with ivermectin aglycone, a derivative of ivermectin, which is missing this disaccharide group, to analyze whether this group can have an impact on the susceptibility or not. So in summary, we're able to show for the first time a direct influence of a hemoncocib on macrocyclic lactone susceptibility, so specifically for ivermectin, and confirm thereby the so far reported differences between ivermectin and moxidectin in terms of pharmacokinetics. And to further analyze the ML substrate specificity of this specific hemoncocib, I'm testing for the macrocyclic lactones. So besides ivermectin aglycone, I'm also testing doramectin, selamectin, and eprinomectin. In addition, we're also comparing the genomic sequence of this specific CYP between different susceptible but also resistant isolates to identify polymorphisms potentially associated with ML resistance. So, and that's it. And I'm thankful for the attention, and I'm happy to take your questions as well as comments and hints for my further work. And I would like to thank Dr. Jürgen Krücken, he's my supervisor, and also DFG for funding.
Thank you very much, Nathalie. So I see no questions from the Mentimeter, but my comment is that your work is very interesting and very important because we need genetic markers for macrocyclic lactones. Oh, there is a question. Do you know if there are any nucleotide amino acid difference between the WR and the Assay strains in Hemoncus contortus, or just expression differences?
in respect or in regard to this
Yes.
genome testing, okay, so this is what I would like to do next. So I sequenced the subsequence from genomic DNA so far, and I was able to see so I sequenced it for ISE. For McMaster isolate, we selected at our institute, so a highly resistant isolate and also White River. And what I can say so far is that there are differences. So for the genomic sequence, but yeah, I know that we need to perform also additional, for example, qPCR studies between different isolates to say whether there is a difference in constitutive expression or if there are really single nucleotide polymorphisms that can explain this effect.
Okay, thank you very much. Thank you. Johannes, you can take over for the poster presentation. Thank you.
Yeah, everybody for the poster has 1 minute. And then the next speaker can immediately take over.
Awesome, great. All right, hello everyone. So I'll try to be brief as possible. So in this study, we want to evaluate the reported levamisole resistance marker ACR8B in Haemonchus contortus. So briefly speaking, we used adult worms from 3 phenotypically resistant, 3 phenotypically susceptible, and 3 Swedish susceptible to levamisole isolates, as well as larvae from these Swedish farms where levamisole was considered to be effective. Next slide, please. All right, so essentially what we did, we created a ddPCR assay, and we found that the 63 base pair deletion that happens in the ACR8 gene is actually way more common overall than not. And even though statistically speaking, there's a genotype difference, and these are significant between the susceptible and resistant isolates on rows 1 and 2 on the left, the ones that are marked S and R. But the deletion containing allele is overly abundant, even in susceptible isolates, essentially. Now, in the larvae on the right hand side of the screen, different frequencies of the deletion containing allele were found in the pretreatment samples. However, the treatment was effective nevertheless, as measured by the fecal egg counts, as well as ddPCR. So overall, this casts some doubts about the utility of this deletion as a resistance to levamisole marker in the field context. Thank you very much.
Okay, thank you for the opportunity to present my project. My name is Paula Ehnert from the Freie Universität Berlin. In attribution to recent studies on enthalmentic resistance in Europe, we look closer on the current resistance situation in northeastern Germany in Brandenburg. And we can provide initial findings of a fecal egg count reduction test that has been conducted on 11 sheep farms using the drugs fenbendazole, ivermectin, and moxidectin. Results were achieved using mini-FlowTag with a 5 eggs per gram sensitivity and analyzed using the R package egg counts. Results can be seen on the second slide of my presentation. Reduced efficiency was found for fenbendazole and ivermectin on each 6 of 8 farms tested, plus 1 farm with suspected ivermectin resistance, while moxidectin resistance was detected on 2 of 11 farms. The perspective of our study is to do the same fecal egg count reduction test on cattle using fenbendazole and ivermectin in spring 2021. And the collected material will be used to conduct a larval migration inhibition assay using levamisole and to determine nematode communities before and after treatment using the Nemabome next generation sequencing approach. Thank you very much for your attention.
Hi to everyone. And this study was performed in collaboration with Ghent University, and the aim was to perform a first validation of the cubic FlowTag microscope on fecal egg count of gastrointestinal nematodes in cattle. You know that the KFM is a new digital microscope that combines high sensitivity, accuracy, and the precision on FlowTag and FlowTag techniques with a label system to capture and analyze the pictures. Please, the second slide. In this study, we collected 30 positive fecal samples from cattle experimentally infected by genes in Ghent, and in lab, we have prepared 6 subsamples for each fecal samples, and we have used 2 different reading systems, a traditional optical microscope and the KFM. And the results showed a substantial agreement with a low discrepancy, as you can see in these 2 graphs. And the KFM is a promising tool for an accurate and efficient fecal count and to perform a remote diagnosis as expected by teleparasitology. Thank you.
Good afternoon, everyone. I am Marieke van Akke from Ghent University, and I've been doing some research into risk factors for infections with Dictyococcus viviparus in dairy cattle and also in associated production losses on Flemish dairy farms. So what we found is that farms have a high risk for an outbreak when the bulk tank milk ODR is above the cutoff of 0.41, especially when this is the case in 2 consecutive samples and in August or October. Also, the risk for 2 consecutive positive bulk tank milk samples was higher when a farmer frequently purchased new animals and when a higher percentage of the grazing season was covered by enthalmentic treatments. The risk was lower when at least 50% of the pastures was mowed, comparing to not mowing at all. Second slide, please. Linking each test day of the milk production to the closest bulk tank milk sample, we found a loss in the mean milk production of 0.26 kilograms per cow per day, and a loss in the mean percentage of fat of 0.02% over the interquartile range. There was also a loss in mean milk production, mean percentage of fat and protein when farmers had 1 or at least 2 consecutive bulk milk samples above the cutoff of 0.41 ODR. Thank you.
And now we move, I cannot share my screen, but it's okay. We move forward to the moderated discussion. We have 4 speakers. The first is Euron Thomas, European Operations Manager of FECPAC. And so, Euron, I have a question for you. As I mentioned, it's very important to sell our diagnostics to the farmers. So, what are your opinion and what are the benefits and limitation to stress when promoting fecal egg counts?
Yes, good afternoon, everybody. Yeah, the selling of fecal egg counting to the farmers to persuade them to pick up stool samples, fecal samples from the field is difficult. But, you know, there are many factors in terms of the benefits. One of the main ones, you know, in terms of, you know, what we talk about in how we do sell to farmers is the resistance story, obviously the enthalmentic resistance. However, I think we can be guilty of overdoing that story sometimes because it's a kind of negative story, the resistance, so on. So we do, and I've been guilty of doing this myself. So there's definitely the resistance and avoiding resistance, but there's lots of other benefits to talk about. So I mentioned, even if there was no resistance at all, monitoring what is happening by fecal egg counts or other means is actually still very important for farmers. So we talk about targeting treatment to when it's needed, and the responsible use of anthelmintics, so the responsible use of medicines is becoming really important. And the effect of using responsibly, targeting treatments which you know are working to the right animals at the right time, and looking at the performance benefits. So we've got to measure the performance benefits of being able to target the correct treatment at the right time, which we are doing, but it is difficult to do on a commercial farming enterprise. And I think the other parts as well, you know, is the environmental story. Sort of great work done by in Scotland by SIUC and Morden on the emissions side. So the effect of parasitism on emissions and so on, which is obviously becoming a very important story in how food is produced. I think we need to be utilizing them, but there's the other side of environmental is the effects that the drugs, the chemicals are having in the soil when it comes out in the dung, so what effect it has on the dung and soil fauna. So I think it's really important to talk about the benefits sort of as a whole, and there's multiple factors there. Limitations, you mentioned, Laura, as well. As we know, limitations, there are a lot of limitations with fecal egg count, and it's important that we are open about that and we do discuss those. So it's the fact that we only pick up adults. Fecal egg counting only picks up adults, which at certain times of the year are very important. So we talk very openly to our clients and customers about that. We run a webinar early in the year, and that was one of the hot topics. But also the variation within tests and between tests. So, you know, people get excited, our clients get excited when they see a difference between 200 and 300 eggs per gram, but a lot of it is the education, if you went, and the backup service to how we promote fecal egg counting and then understanding that and the combinations. And I think one of the key things for me is when we're selling fecal egg counting is we often look at fecal egg counting as a diagnostic, which it is a diagnostic, but sometimes we only think of it as a diagnostic and as a one-off kind of every now and again test. Where we see the benefits of fecal egg counting and what we try and sell is the regular monitoring. So fecal egg counting should be done on a regular basis throughout the season. So our best the customers that we have who are convinced with fecal egg counting are those who are doing 30, 40, 50 tests a year or more. So they are monitoring different groups of animals separately every time, and then doing that on a regular basis. So it's about tracking trends and also building up a picture. It's being data-driven, building up data, what's happening on farm, being able to share that with your advisor, veterinary advisor, or so on, and using that in a pattern. So I think we need to really push fecal egg counting as a regular monitoring tool, which is common practice on farms, rather than if you went one-off diagnostics, I think.
Yeah, and we have some questions. Should enthalmentics be prescription only in response to high fecal egg counts or not?
I think the argument, yeah, it's interesting. There are parts of Europe now which we know are going to prescription only. It would be nice to not have to go down that. You know, it'd be nice to actually have a kind of voluntary uptake from farms. But it looks as if, you know, sort of we are going down that track. I know in Ireland, this is kind of an upcoming thing. So yeah, whether it should be, there's a bit of a political debate. You know, I think we do need more care around how enthalmentics are sold. And only based around fecal high, I think you've got to be careful only around high fecal egg counts. I think fecal egg counts backing it up, but I think we have to be careful because we know sometimes we need to go in with a treatment, Laura, as you know, at low, nematovirus early in the season, we might have to do it. We saw an issue after our drought broke this year in the summer where we had a really big explosion of worms. So I think we've got to be careful only relying on fecal egg counts, but it's using data and information, which fecal egg count is a part of that.
Yes, and this especially is true for cattle, no? Where the low fecal egg count. Yeah,
absolutely, yeah.
Okay, thank you very much, Udo. We move on. Menno Olzauer is a scientist and consultant at Royal GD Deventer, the Netherlands. Menno?
Yes, good afternoon.
How are you?
Fine? Yes, I'm fine, thank you.
Thank you for your participation. So,
okay.
How can vets be involved in diagnostic services in parasitology, according to your experience?
Yeah. To my experience, the role of the veterinarian, of the practitioner, is to advise the herdsman to monitor the herd for parasitic infections and to advise the herdsman for parasitic infections properly and advise the farmer for treatment, for metaphylactic treatment and preventive vaccination, and also to monitor the correct effect of treatment. So in our country, I think the first part of the answer is that is, correct done, but monitoring is not very much done in practice. So that might be better. And then the challenge for the veterinarian is, in case of problems, is of course first to select the right animals for examination and to take the right samples for additional diagnostics, and the therapy applied and the correct expectation management. That's also very important to predict what the herdsman can expect. And I think, as we all know, the epidemiology of some parasitological infections is complex. So to be able to optimal advise the dairy farmer and proper handling of pharmaceuticals good knowledge of this epidemiology is important.
So our role is to disseminate our knowledge to farmers. Maybe they should be educated regarding parasites.
Yes, absolutely. And that's all what also part of our work in our country to train the practitioners in taking the right samples and corrected advising of the dairy farmers.
Okay, thank you very much.
Okay.
Menno. Thank you. Okay.
Okay, you're welcome.
And now our expert, my co-leader and friend, and our expert on molecular diagnostics, Professor George von Samson in Mestierna from the University of Berlin. So, George, most of the presentation today were on molecular biology. And what do you think? What kind of new diagnostics are in the pipeline for the monitoring enthalmentic resistance from a molecular point of view?
Yeah, from the molecular point of view, I think we still have the problem that there's only basically one drug class. Now, after the talk of Cedric, we heard there's also some data, and that is the benzimidazoles. Now, after the talk from Cedric, we all have a nice impression again on how is the situation on molecular markers in nicotinic acetylcholine receptor acting compounds. There is some evidence for some markers there, but there, and also concerning the benzimidazoles, the recent findings published rather illustrate that there is a greater variety of polymorphisms in the field, also with relevance in different geographical regions, but maybe also in the same region in different populations in the beta-tubulin sequences for BZ resistance. So what we sometime many years ago initially thought it's a codon 200, we know for long that's not the case. We know that there's also 167, 198, but now we also know in 198, for example, there are different amino acids which are changes which are associated with resistance. So it's becoming more and more complex. And that also limits a universal diagnostic tool. Optimally, of course, we would have a easy next generation sequencing test. But at least to date, this is, I think, not there that we can imagine this in the short term frame to become available for routine diagnostics. So I think we are a little bit, we have to get back into the science, and it will take more efforts to develop this. In this respect, I find it what we also saw from Cedric's presentation, then also relevant to discuss testing on non-molecular basis with a more or less versatile multi-species, if that's possible, phenotypic test, in vitro test. Yeah, if that is giving you also, and that is of course immediately and much more directly giving you an impression of the susceptibility in the animals as well. If that is feasible, that's of course also quite or very much attractive, I think. And
you think that milk ELISA is still a good tool to be used?
The milk ELISA, yes. I think it's a good tool to target treatment to herds which have a problem, not on the individual animal basis, but as we've seen in the talk before, there is this association, I think, still, I believe it is there. And we also see it that the effect on animals treated, that's a study where we were involved with primoparous animals which were treated, then concerning milk production was increased only in herds with high ODR levels. So I think to identify herds being in where of benefit of enthalmentic treatment is a good application of this tool.
Okay, thank you very much.
But maybe to add for the macrocyclic lactones, which are of course very, very important, we are way behind, or we're still lacking a lot of information, and we don't have even one slightly universally accepted or occurring molecular marker there, but that's still a big working place there.
But you are doing very well at your lab, so I'm sure that there will be some news in the pipeline from your lab.
I think the problem we are facing is the variability of the parasitic nematodes. Yeah, they have such a big genetic versatility and flexibility that maybe in the end, on different geographical regions, they'll always be able to evolve different specific mechanisms. And that'll be the challenge for the future, to deal with that.
Thank you very much, George. And our final expert is my lovely friend Smaro Sotiraki from Greece. So in the past, we saw that Italy was a happy land for enthalmentic resistance, and maybe even Greece. So Southern Europe didn't face so much problems as Central and Northern Europe. Can you comment on the different situation in Europe, and also if we need different approaches to tackle enthalmentic resistance across Europe?
Good evening on my side. And of course, as you mentioned earlier with Hannah's paper that came out recently, we also saw that there was also lack of evidence. And exactly as you said earlier, Laura, if you search, you find. So it has to do also with the research effort in different areas. And of course, that it's one of the problems. And depending how organized the farmers are, how much they look for resistance, how well educated they are. And this is also in some eastern areas of Europe that it's lack of information. And of course, the management of and the type of husbandry systems. For example, if we are mostly dedicated to milk and we have some limitations in which drugs we are using, then it means that we do not apply as many treatments per year as they do in Central and Northern Europe. And of course, it's the mentality of the farmers. I think if in countries that there are more sheep and goats and they are small farmers that they are not so professional, they don't really follow instructions, so they don't do regular treatments, whereas in other countries they are more, I don't know, they comply more with what has to be done. And I'm coming back to Julian's comments earlier that education, I think, is very important. So I wouldn't say that if there is need for different diagnostic approaches, is who applies what and how they are educated. I could never think that the farmers would ask themselves for testing, fecal count testing, and in Greece, and I would not expect also the vets to start doing fecal counts themselves. So it has to come from specialized laboratories. And to be honest, we have programs that they are driven from pharmaceutical companies that they actually offer to the vets for free, that we do fecal accounts, and even the vets are not so willing to go to the farm and collect. They prefer just to sell drugs and with not really a management scheme, and especially if the farmer is not so professional to be able to pay.
Yeah. Indeed, from the audience, how likely is it that farmers should adopt molecular diagnostics if they are not willing to go for fecal count?
Yeah, so we have to start from the basics first, and I think we start from education and what we expect, and that's, I think, the more important thing. And it has to do with the professional level, and it's different even with our countries. I think with the southern part. I think cattle farmers are more professional, and of course we don't have problem with cattle because the cattle are mainly indoors. We don't have the pastures and the ability due to the climate to graze outside. So that means that we don't have any problem with cattle. That doesn't mean that we use the enthalmetics correctly. But if the farmers are not that professional and they don't really go into other basic things, then parasites comes as a second, I think, priority.
Yeah, and also maybe we need new approaches because there is a high genetic diversity in the population of helminths, and this might confound also the enthalmetic resistance status, because yes, especially in Southern Europe, we have these polyparasites with several genus and species.
And also the climatic effect that we don't know how it affects the resilience and the resistance of the parasites.
Irion, there is another question for you. How many subscribers you have at the moment?
That's a question I hadn't prepared for. I should look up before. Between our subscribers using mic's on, isn't it? Yeah. Between our subscribers using our systems on farm, and it's not only that, we actually use veterinary practices offering a service using our system, as well as retail stores using the system. So we have a number of farmers using them. You know, we will have probably, I think it's about 500-600 users between the on-farm and people subscribing through vets and stuff using our sort of whether vets are using our systems. This is in the UK alone, so there's still a long way to go sort of on that. I want to probably add something into what Smaro and Menno mentioned in terms of the vet kind of role in this, and the veterinary role, where we have really good interaction and uptake is where there is a really good relationship between the veterinarian or the animal health advisor, so the people selling the wormers, they have a good relationship with the farmer, and they work with them for proper health planning, and that does work very well. And as Smaro mentioned, maybe, you know, that the vets prefer to sell the product and the enthalmentic and so on. I think we're very lucky here in the UK. We have SCOPS as a big industry body, which is doing a massive amount of educating the veterinarian and the people who are selling the wormers over the counter. There's good education actually of those to help engage farmers in doing more of this. But yeah, we still, you know, we get good inroads, but there's a long way to go still.
Yeah. Okay, thank you very much to all the panel members. And now Johannes would like to show the Mentimeter results.
Okay, this was the first question shared with you. In fact, if you didn't yet answer, you can still enter. So go to menti.com with the code, enter responses. So from which country are you dialing in? As you can see, from a lot of countries. And I also learned that Wales and Scotland and UK are all different countries. So that's learning experience for me. But so we also see from Nigeria, Brazil, etc., but most from Europe. So let's try to go to the next slide. So still there are still some answers are coming in, so I will wait maybe a little bit. Because we are 100. Yes, of course we are at the time. I hope some of you, so this will only take, going through the questions, maximum, I think 10 minutes. So then we should really be able to close this meeting. I hope you can still stay with us until then. So maybe I'll wait until we have about 120 responses or something, or we'll see how quick it goes. How quick the votes go up. Okay, but anyway, so keep on answering. We will keep the records also for our report. But so the main message is clear here. A lot of different countries and a lot of Europe, as we of course expected. So I'll go to the next question. In your perception, is enthalmentic resistance on the rise in Europe? Yes, of course, this meeting is on this topic, but there's a clear picture on this, that a majority thinks that yes, it's on the rise in Europe. And so, well, this is of course not good news, but it's a job for us to work on this and make sure that in the future the problem becomes smaller instead of greater. So please, you can still continue to vote even if after if I changed the slides, because the more inputs we have, the better. Then, okay, this is probably a difficult one. What kind of diagnosis is best suited to underpin sustainable parasite control? Okay, I can scroll. So there's a lot of different inputs, both fecal egg count reduction test and molecular markers. Also same minifloat and PCR. Combination, I see. Fecal egg count reduction tests, combination. So we will have a good look at the different comments received, and hopefully we can include the general picture in our meeting report. So there were 37 inputs. I think even after the meeting, I'm not sure, but I think it should be open, the question, even for a few days after the meeting, so you may still be able to provide some inputs. And I see a lot of the same tests, combination of both, let's say, phenotypic tests, as Georg said, and molecular markers. Okay, so I go to the next slide. And I think this is the last one. What are the main diagnostic needs for assessment of enthalmentic efficacy in the field? So if we read the biggest words, these are the words that are most frequently reported: reliable, fecal egg count reduction tests, Parasite, quick results, inexpensive, user-friendly. So for yes, these are the keywords. We more or less know these. So we need to keep on working on these keywords so that our results. First, we need to have tests. We learned from this meeting that in a lot of areas we still need tests. There are no practical tests yet. And once we have tests, yes, we have to work on cost effectiveness, on reliability, on quick results, etc.
So maybe, Jannes, when Combar meeting is over, we can transform these charts into slides.
Yes.
Like, yeah.
Yes, definitely.
And the first question was, will the slides be shared? The slides from the speakers?
Well, I think what we definitely will do, we recorded the meetings and we will share the recordings of the presentation. And of course, in the recordings, there are all the slides as well. I think this is a more clean approach. We can put it online rather than put all the individual presentations online. That is what we will do. But this will take some time. In the meantime, yes, I will close it now, but you may still continue to provide inputs.
Yes, I think there are no more question. Talking about diagnostic standardization, realistic, complicated protocols are likely not well received. Yes, indeed, as I mentioned, we are working on the fecal egg count reduction test protocols. It's just 2 pages, but we received so many input, and this is why we are going to discuss this online in January. And of course, standardization is an achievement, is a goal not easy to do. And especially we have to consider that sometimes research purposes are different from practical ones. So maybe we can find a compromise between research and practice.